right, good afternoon everybody, and thanks for coming to the DeFord lecture this afternoon. We're very lucky to have Bruce Fouke coming down from the University of Illinois. Some of you know him already, some of you know him really well, but we're very happy he made it here. Over the last few weeks, I've discovered that I think I may be the only person I know who doesn't do planetary geology. So I'm starting to discover I need to step up my game here, because I'm okay with the Bahamas, go to Mars, it's kind of dry. But nonetheless, I need to get modern. But Bruce has had a remarkable career. Grew up in Iowa, then went to Bradley University, did a master's at the University of Iowa, which has got some great stories associated with it, as well as the University of Chicago, and then did his PhD at SUNY Stony Brook, where he met Jay Banner here. Did a few postdocs, not just one. He did a postdoc at Vrije University in Amsterdam with Wolfgang Schlager, who that's an experience for a career in and of itself, and then at Cal Berkeley, famously working there with Walter Alvarez, and then with the exobiology NASA Ames Research Center, so just a huge experience there. And then he's been at the University of Illinois for a number of years as a professor in the Geosciences department, but then he's also kind of spread out his footprint across the University of Illinois, so he works there as the Ralph Grimm Professor in the Department of Geological Sciences, or the Department of Geology. That's the topic these days because we're talking about changing our name.
Yep.
So the Department of Geology, you still are. Right?
We still are.
Okay. But he's also associated with the Carl Woese Institute of Genomic Biology, and then he's the director of this amazing Illinois Roy Carver Biotechnology Center, which has the most remarkable spectrum of different analytical equipment, from every kind of microscope known to man to every kind of genomic technology. So quite an experience. And so he's involved also with the chemistry group now too, or biology?
Yeah, both. So.
Anyway, so kind of like a lot of, if you guys multitask in different groups, everybody always said, well, where's so-and-so? I haven't seen him in weeks. He doesn't work for us. So he's always wondering in the geology department where Bruce is. But he's working on quite a few things. And today he's going to talk to us about, again, bringing in some of these different technologies to look at things that we would call earth science problems to an extent, but they're also crossing the hall into the medical arena. And a lot of us here might think about that, because certainly as far as funding from the university goes, and our AD of research can tell us, but aligning yourself with Dell Medical is not a bad idea if you want to get research. So some of the ideas Bruce presents may bring up some thoughts for you for the different types of research you do. But I'm looking forward to that talk.
Charlie, thank you very much. So it's such a pleasure to be here today. And I've worked and known Charlie, great friend, great colleague, for 30 years. And Charlie, thank you. And Charlie set the gold standard for understanding how to interpret and predict the dynamics of very complex marine systems. And anyway, thanks for all you forged. And I was looking at my notes, and remarkably, today's November 10th. It turns out that 40 years ago today, I was a graduate student at the University of Iowa, and I went through the library and started looking at marine carbonates. And I kept seeing this name, Lytton Land. And so I never met him. I'd never been to Texas at that point. And so I had the courage or something I wrote to Lytton out of the blue. And I still have the letter. And it said, Dear Professor Land, I grew up in northern rural Iowa. I can farm, I can weld, I can scuba dive, and I want to study marine carbonates. And Lytton, in classic Lytton style, he took the letter, he circled the words scuba, and he circled the word welding, and he drew 2 lines, and he just put Jamaica this Christmas, question. So I called him, and the next thing I knew, I spent the month of January with Lytton and Judy Lang. And that's where he walked me through the Falmouth and the Hope Gate and all the Dolomite models, and it started a lifelong career trajectory working with Lytton and Bob and everybody. And then Jay and I were both a product of Stony Brook, another great place for marine and all kinds of terrestrial carbonate work. And so I've had 40 years of very deeply appreciated interactions with your department. So thank you so much for that. It's been a major part of my ability to contribute across the board. And again, thank you for the very kind introduction. What I want to do is kind of open the door even a little bit more and talk about some of the horizons that we can look at in terms of geosciences. And I should just want to say too, congratulations, #1 rating in the US. That's pretty amazing. And I'm here too, the DeFord is to get you jacked up. Oh yeah, that's what I'm talking about. So we have to get all of you grad students and postdocs engaged, right? Because you're at the #1 program of the US. You're working in the centerpiece, the foci of the future of science, engineering, medicine, technology, environment, energy, space exploration, human health. You are in the middle of that. How exciting. And congratulations on making such a great choice. So I'm just here to kind of further encourage you to go forward and realize what a great choice you've made to be here at Texas. So another thing I want to point out is that I was asked by the National Park Service to write a book about our 30 years of work in Yellowstone. And I wrote it with Tom Murphy. If you've never seen Tom Murphy's work, just Google Tom Murphy photography. It'll pop right up. But if you're interested in a free copy of the book I wrote as part of the 150th anniversary, all you have to do is Google NASA Art of Yellowstone Science. And you can, oh, I'm sorry, I forgot to do that. That's my fault. And so you can actually download a free copy of the book if you're interested. But there's a lot of science in there. It goes from the science of biomarkers, travertine deposition, water rock interaction, hydrodynamics, space exploration, but we also go all the way through to human medicine, the human history of Yellowstone, all kinds of things, and we have 260 of our favorite photographs that Tom Murphy and I have taken over many years out in the wilderness. So please feel free to do that. That's the NASA website, NASA Art of Yellowstone Science. All right, so what are we going to talk about today? Well, it's always interesting, I think, as most of us know, to work on something that no one really knows what to call what you do. And that means we're actually, I think, doing some pretty good science, right? So geobiology, astrobiology, universal biomineralization. This is the concept that life originally had to deal with mineralization in the environments in which life first emerged, first evolved. And then life evolved, adapted to the point where it could harness that mineralization to the benefit of the organism surviving the environment and being successful evolutionarily, right? So that's what I'm calling all these processes of biomineralization. And then in the literature over the last couple of years, I've tried to establish something I'm calling Geobiomed. And I wanted you to know, I know it's always crazy with terminology. We worked for years to get this word, right? So we worked with the doctors at Mayo Clinic, Northwestern, UCLA Health, all over the US. And then we worked with all kinds of scientists. And we actually came up with this phrase because the word geohealth, that's been used somewhat by GSA, but it's also, geohealth has been captured by the geography group, right? And that's where you kind of correlate disease distributions with different, geographies and climates, which is excellent work. So we also wanted to not, try to hijack someone else's term. So Geobiomed is the idea of bringing together the cutting edge of biology, geology, and medicine together to answer hugely important questions within the medical field. Now, I just wanted to point out that all of us can understand, I think, fundamentally, if we look at some of the models for the emergence of life at about 3.8+ billion years ago, these are environments in the deep sea floor. Here's the Smithsonian showing the Alvin going down to the Lost City. And I just want to point out, those are dominantly, the Lost City are carbonates, and you could actually call those travertine. And some of them are not so different from what we find at Mammoth Hot Springs in Yellowstone. But this is actually a diagram that I put together with Carl Woese, W-O-E-S-E. He was a professor at Illinois and someone who I worked with for a long time. He's the one who revolutionized, on November 3rd of 1977, he published a paper in PNAS that proved that we have 3 domains of life, as we show in this diagram: bacteria, archaea, and eukarya. Not five kingdoms, right? And everything did not just evolve from single cells to multiple cells, right? So this is the map now, the roadmap based on the 16S ribosomal RNA gene of the, so if you ever get everyone's from Earth in here? Okay, good. And how many people spent time with, in spacecraft, aliens? There's just a couple in the back. Okay. And so if you ever get captured or taken up to a spacecraft by an alien, and one of the questions, you're an Earthling, how many types of life are on planet Earth? For goodness sake, say 3, right? Bacteria, archaea, and eukarya. And remember, the archaea are single cells, they're the methane producers, and they're actually more closely related to humans than the bacteria. Okay, so the archaea evolved long after. We have Luca, the last universal common ancestor, and then we have the last universal eukarya archaea ancestor, all right? So just because it's single cell doesn't mean it's simple, and it doesn't mean it's more related. And in fact, our genome with the archaea overlaps a lot. So just, these are perspectives we need to have. And on that deep sea floor, where we think life evolved in these thermal fields, these thermal vents, you always had mineralization. It was from the very get-go where we go from RNA-based life to the first protocells that were forming on the seafloor. And in those environments, mineralization was happening. So I'm trying to just reset the stage. We should look at mineralization. It should just be something that flows off our tongue when we say life, right? Because life and mineralization, they can't be disconnected. And they're intimately intertwined since the dawn of time. So what I've tried to do over the last several years, and again with a lot of help from great colleagues here at Texas, among other places, is I've looked at this as an immense triangle, right? Life, water, mineral. And I've set about, through great colleagueship and then trying to learn the most I could too, to quantify each of those apices of that triangle. Physiology, metabolism, ecology, and evolution for life, and use the whole suite of omics technologies. Mineralogy, so it's the chemistry, the mineralogy, the sedimentology, the stratigraphy, the diagenesis. The water, the hydrodynamics, the chemistry, the transport. And then putting all those things together in what I call contextualization and integration. That's one of the things we do as the Geosciences that is untouched in other disciplines, time and space. And that context allows us to then to move in, do quantitative modeling, and then what I've been doing with Charlie Worth here at Texas in the environmental engineering program is to be able to do microfluidic testing. Now, I wanted to know what to call this, right? And I was trying to think about it, and I was doing field work and lecturing in northern India in a place called Dehradun. And it's Rudyard Kipling actually was there, and that's where he wrote a lot of the Just So stories. And it's an incredible place if you ever get to go. And we went up into the mountain stations up in the Himalayas. But the thing was that I was standing in front of the statue of Lord Ganesha, which is the supreme god. And at that moment, I was trying to think it through, and it became clear. I mean, it was just like lightning hit me. The eternal triangle. So you'll have to give me the poetic justice. The eternal triangle is what we're going to call this. Quantifying those aspects of life, mineral, and water, so we can answer these really difficult questions about life in the past, life in the present, and life in the future. So here's now I kind of want to reset. I'm not sure if you really understood this or not, but Urbana Champaign is actually in the center of the universe. Okay, so this kind of shows the distribution of, I mean, for those of you who've been there, you can understand, it's one of the vacation capitals of the entire world. And so I've been very fortunate to be at Illinois, and we have a great little airport. So, but these are the places that we've worked over the years. And these are the manifestations for our group of the Eternal Triangle. So we've done oil and gas exploration, deep subsurface, the whole gig of exploration, basin fill, migration, all those kind of things put together with studying the microbes that live in the deep subsurface. Then we've had Yellowstone. Let's see now. This is a pointer, right? I'm not blind myself. Okay. So there's the North Sea of other places, but we've also worked in the North Slope of Alaska and what have you. Worked a lot in Yellowstone looking at water, life, mineral interactions that make biomarkers. We had a NASA Astrobiology Institute grant, and then the idea was to use that now as a template to help with the search for fossilized life on other planets. I'm going to talk to you about our work at the Mayo Clinic, Northwestern, UCLA, understanding how we can take principles from hot springs, coral reefs, and the deep subsurface and understand biomineralization in the human body. We've worked a lot on corals. We looked at coral disease, the genesis of coral reef ecosystems as they're fossilized in marine carbonate systems tracts. It's just, the whole discipline, putting it together, but then bringing in things like RNA, DNA, protein sequencing into the middle of that We've also worked a lot in Rome on the Amio Novus aqueduct. The aqueducts began life, water, mineral interaction, and the aqueducts, as they were flowing, they precipitated calcium carbonate in the form of travertine. And then there's a picture of Kyle and I here on the Great Barrier Reef, collecting samples of coral skeleton and tissue to look at the genome of the coral, the expression of genes, how those genes and proteins are used to attenuate the growth rate of the coral, and then I put that into a climate change understanding of, as sea level changes and temperature changes, what can we expect from the corals. And so there are just three people I wanted to mention briefly, and you're going to hear a little bit more about them later on. But one is Dr. Mayundi Sivaguru. We call him Shiv, and he's been here several times in your labs, coming down here from Illinois. And he's a plant biologist and someone who's worked with me on all these projects. I'd already mentioned Dr. Charlie Worth, who is an expert geochemist for carbonates, and also his main technology kind of centerpiece is microfluidics. So we built something that he wanted to call it a microfluidic device, and that just wasn't too sexy. So in the literature, we call it in APG Bulletin the Geobiocell. What do you think? Would you want to buy one? I got a couple right here. No, so the names, right? So the Geobiocell microfluidic device, it allows us to test life, water, mineral interactions. And then I just wanted to thank Kyle, who's worked with me over the years, and I'm just delighted he's here working with Charlie and the rest of you. So, and I just wanted to mention too that I've had over 50 graduate students and postdocs over the years, and everything I'm going to show you is a result of these incredible, I tried to make a slide with everyone's name, and it just, it looked like looking at the front page of the New York Times from 30 feet away. So it just wasn't working, but I just want to express my appreciation to a very large cadre of people who have made all this happen. And also, of course, the funding agencies across the board. So now I'm going to break my talk in just a couple sections. The first one I want to talk about is what I call philosophical and technological toolkits. And again, when you get a chance to have the honor, and again, thank you for the invitation for the DeFord. It really means a lot to me. Thank you. And you want to also have it be a teaching opportunity for all these brilliant superstars called graduate students and postdocs sitting in the audience. So I just want to tell a couple things which I know it goes without saying at one level, and another level it's kind of important to say. One of them is my vision of scientific method. And of course, scientific method is the centerpiece of what we do as scientists, but I actually like to modify it and call it scientific inquiry, where I see it more like a giant swirling tornado, and truth is what we're after, and it's a translucent thread that runs through the middle of the tornado. And so, what I have here is you have curiosity, reasoning, hypotheses, experimentation, synthesis, discussion, results, communicate results, and then reinitiate your curiosity. And this is looking at the tornado from the side, and this is looking at the scientific inquiry tornado from the top. And good science, it puts you closer to truth. And then you can tighten and tighten and tighten, and what just happened? There we go. As we go through time. And so that's how I see the scientific method. Actually, to tell you the truth, when I was in rural Iowa, and I don't know why this is blinking off and on. It's an omen. Let's see here. Now we're going to do an Indonesian shadow puppet. This is what beer comes out. Everyone. We're done. Truth and beer have been found. Not in that order. Not in that order. So.
Have we lost the bulb?
Wow. That is how excited that the whole system got. Well. See, working with Charlie, there's always a test, right?
You want to reboot it? You want to see if that'll wake things up?
Well, I did that.
Boy, that's a shitty connection. That can't be a connection, is it?
Yeah.
Is that? God.
Let's try to plug this into...
That's the HDMI. Just plug it straight in.
Jay, how you doing? It's good to see you, my friend.
If you really lost the bulb on the projector,
Then, yeah.
nothing will work. What you
It's
could
over.
do, no, is you could try to use the overhead presenter and put your screen flat.
Oh, that's a good idea.
Is this? I'll just get this ready.
Let's go to Plan B here. So.
Bruce, where are your adapters?
Yeah, right here.
I mean, there's an HDMI port. We don't need an
Yep.
adapter. We just put it straight in.
And just put it in straight.
Just in case it's
Adapters here. Okay, unplugging that. Bring this over
And
here.
that might have been easier in the first place.
Yeah.
I can't see, can you
I
plug
got
it
it,
in?
yeah.
But if it's the bulb, I don't know if it's the...
Yeah, it was the bulb then.
Oh,
Oh, woo! Charlie, thank you. All right. Wow, thank you. Okay, so let's see, where was I? I was just trying to say that this is my version of this. When I was trying to learn scientific method in high school, I really got disinterested in how it was taught, and it was a very linear kind of experience. And so this just made a lot more sense for how science worked for me. Another thing I just wanted to point out that the powers of 10 of space and time, how big, how small, how old, how young. I try to put all of our projects into that contextual framework before we start. So this is just a snapshot of our work for Yellowstone. I know there's a lot here. What I want to point out is on the y-axis, this is 10 to the negative 9th, 1 nanometer, going all the way up to 10 to the 5th meters. And up here is a supervolcano. Down here is the size of a microbial cell and some of our abilities to detect using different instrumentation. Here's field work and observations and sampling. Here's laboratory analyses. And this incredibly important linkage at that kind of millimeter to centimeter scale, making sure that we have data sets that allow us to cover this range quantitatively. So it's just putting things into a powers of 10 framework. And we've done that for the human body. I'll talk about coral reefs, Roman aqueducts, caves, hot springs. That consistency in doing that has allowed us to do the upscale modeling to make predictions. And it's an exercise you have to do at the beginning of your work, not at the end, after all the data is collected. And this allows you to collect the right kind of data at the right scales of time and space. And then another thing that's been so important, I just want to point out, again, the geographic significance of Urbana Champaign. We're only 32 hours from Yellowstone driving. I mean, just think how convenient that is. It's incredible, right? So here we are working in Yellowstone, and what's so important about it is that we do integrated lab and field, and we go lab, field, back and forth, back and forth. And our permits in this one, we put siphons down the vents. Of course, we work very closely with the Park Service. And then running these controlled experiments allowed us to quantify the effects of proteins produced by microbes to actually increase the growth rate of the carbonates that grow. And then Kyle and I were diving in Australia, and this is a deep dive where we were taking cores. And again, we were working with the Australian Great Barrier Reef rangers to do this. But back and forth. And that's really, again, these things, I'm just bringing them up because it needs to be part of the planning, right? You can't start doing this at the end of the project. And this is what's really allowed us to make a lot of progress. And then another thing is to harness all the new technology, which you folks are great at doing here. So I'm just bringing the coals of Newcastle, so to speak. So, here's my MD Sivaguru. We call him Shiv. He's been down here a lot with the UTCT. And some of the technologies we're using now are, we just got a grant to receive a new Zeiss LSM 980, and it allows us to do optical analyses, both in terms of bright field transmitted light and autofluorescence or induced fluorescence at the scale of 140 nanometers. So like when young chickadees like Charlie and I were in grad school, we were feeling pretty good if we had microscopes that let us get down to, 5 microns, and anything smaller was just something tiny you called a micrite or a shale because you couldn't look at it. Now, a micrite and a shale is a boulder field at 140 nanometers. So what's happening is all the worlds of microscopy driven by optical electrons, being driven by light, being driven by all forms of energy and X-rays, they're all starting to converge. And it's allowing us to do amazing things. And so here's just a picture of the Stokes shift for the autofluorescence, the organic matter that's trapped in minerals. It doesn't matter what mineral it is. If there's organics, it'll work. And there's a Stokes shift, and you get these very, very characteristic wavelengths that tell you about the organics. And the reason I'm saying this is I'm going to show you a few pictures of this. And then we were so excited by the UTCT that we decided to go home and write a grant and get our own. And so thank you for all the inspiration. We're going to continue to come down here, though, because you guys are the ones who really know what you're doing. And here's Shiv running it, and you're going to see some results of this. So, and then one more thing, I just want to strongly open the door for you. So I've also been the director of the Carver Biotechnology Center in Illinois for 10 years. And we do all things DNA, RNA, and proteins. And we have a staff of about 50 PhDs. We're Illumina High Throughput Center of Excellence. We're a Zeiss Center of Excellence. What I'm trying to say is that we're completely set up to do all the entire spectrum of the future of omics, DNA, RNA, and protein. And we're set up, you don't have to know anything about it. All you got to do is come to us with a sample and talk to us, and we go from there. And so we've opened the door of the power of omics completely to everyone, and we're an open facility. So just keep this in mind. We have counseling sessions at the beginning of projects where people can come to us either knowing a lot or knowing nothing, and then we work with you through the project to add things. And it could be a really important component. So don't feel like because you don't know molecular biology that you can't incorporate it in your project. And then one more thing to say is that we've also just built a new complex called the cytometry and microscopy to omics. The punchline there is that on a scale of 5 microns, if you have a carefully well-prepared thin section, on a 5 micron scale, we can go around and whether it's a tissue or a travertine, we can identify gene expression on a thin section at the scale of 5 microns. So we have incredibly high resolution 2D control on the distribution of this, and it's going to open up all kinds of possibilities for the future. And then also I just want to say, I love to teach, and I always bring in the educational component to everything we do. And I just want to point out one of the important ones. There's everything from Svalbard to Curacao to Yellowstone, what have you. But I also created a massive open online course on the Coursera platform called Emergence of Life. It's free, so if you're interested. And we've now had over 450,000 students globally. And of those, we have all the metrics from everyone who signs in, and 62% of the people of that 450,000 have been from underrepresented populations globally. So it's an incredible tool, and I just want to keep encouraging us to not abandon whatsoever our bread and butter that we do in the classroom, in the laboratory, in the field especially. The field is so critically important. But we also need to remember the power of the online. I'll just mention that I did a scuba dive in Vietnam. I was off in Nha Trang, and no one knew I was there except for the Chancellor of Hanoi and a couple faculty. I came out of the water after the dive, looking like Godzilla, trying not to fall over in the wave action. And suddenly I started hearing out of the blue, Professor Fouke, Professor Fouke. And randomly there were some high school kids on the beach who had taken my class. And they were, I don't know how they recognized me. And they were so excited they ran into the ocean with their clothes on. And so I was trying not to fall on them as I was getting out of the water, and they were asking these great questions about horizontal gene transfer within the hot springs of Yellowstone and how travertine could affect the transfer of genes in microbes. That's the kind of thing that we want to have happen. That's the kind of reach that we can have. Now, these background shots, I just want to point out, this is the super resolution autofluorescence. And you're seeing now pictures where the resolution you're seeing with your eyes are 140 nanometers in thickness. So this is a kidney stone. The link to this across here is something on the order of about 0.5 millimeter. And the bottom line is that doctors had thought that kidney stones don't tell you anything, so they just pulled them out and threw them away. And I said, I've never met a rock, that doesn't tell you a lot of things. And so it turns out that we've now proven that these are all changes in the microbial and human activity of the physiology of the cells in the human kidney. And each one of these layers represents probably something on the order of 1 minute to 10 minutes. Unbelievable. So it's a tape recorder of the physiology of the human being that this stone was taken from at a resolution that we've never seen before. So I'm going to talk about that in just a moment for you, but I think the images are pretty striking. I just wanted to say that the reason that we can, for the rest of the talk, I want to just prepare us to get ready to look at some of the Geobiomed. We've worked in Yellowstone now for 30 years, And again, keep in mind the eternal triangle. The goal was to use Mammoth Hot Springs, which is the only spring out of the 2,500 plus in Yellowstone that actually precipitates carbonate. There's one called Calcite Spring, but it's not really a calcite. It's very confusing. So there's only one spring, Mammoth Hot Spring, and that's where travertine, calcium carbonate in the form of aragonite, and calcite are formed. And so I just wanted to point out that over years, we've done a lot with time-lapse, but this is a time-lapse movie that we took that is over the period of about 10 days. And what you see growing from the hot spring water, the white is the travertine, that's solid rock. And so in 10 days, you're growing that much travertine in 10 days. So that's why we work there, because we have the ability to run real-time experiments. And you know, the graduate student doesn't have to have an experiment that will take 10,000 years, right? Your experiment can take 10 days. And it's just amazing what you can test. And then, you know, we did a lot of photography. We did hydrodynamics. We did the omics. We looked at sulfide oxidation and how that is affecting the water mass flowing over the top. We did all high-resolution petrography. We identified the primary flow path as a function of the dissipation of heat, dt over dr, where r is the radius, and then put the entire story together. And so the point I wanted to make is that this is how we tackled the biomarkers. This work in Yellowstone allowed us to answer similar questions on coral reefs. And then we took the Yellowstone and the coral reef work together, and that's what's allowed us to do the work we've done in Geobiomed. So that's the sequence of events, and it all goes back to the eternal triangle. So now I want to take the rest of the time to focus on two of the Geobiomed areas that we've been working on. And I just want to re-emphasize that you are sitting next to a world-class hospital, right? The Dell Medical Center. And actually, just last week, I was telling David and some others that we regularly have faculty who are physicians come back and forth between University of Illinois and the Dell Medical Center. And so it's just phenomenal people. And so keep in mind, you're sitting next to a gold mine. And what you need, too, the MDs are phenomenal. Thank goodness for them. They're wonderful. But what you really need are the PhD MDs, because they have all that phenomenal knowledge of the medicine, but also they have the understanding of what it takes to get the research done. And you have a really high percentage of PhD MDs at the Dell Medical Center. So also for this one, I will point out, here's our new funding streams for these. And I'm just very appreciative for all these. And I want to point out, you know, Mayanga Sivaguru at Illinois. We have a group at Mayo Clinic. We have the group at UCLA Health. The group at Northwestern, Charlie Worth here. Kyle's been heavily involved, Lauren, Alina, and then Jessica. So that's been our team so far, and we're just scratching the surface on all of this. So a couple things about kidney stones. They affect about 12% of the world's population. 12% out of 7 billion. That's a big number, right? And to really treat kidney stones, we don't have good non-invasive ways. You have to go in and do surgery, or you have to do shockwave lithotripsy. You have to break it up and have it be passed. And none of these are good options. And the average surgery for kidney stones is about $42,000 per surgery. And some of the stones I'm going to show you came from a 56-year-old woman who had diabetes, and it was her 7th surgery for kidney stones. So start doing the math, right? It's astronomical. About 72% of the kidney stones are calcium oxalate, which beautifully, miraculously, and not surprisingly, are very similar to the calcium carbonates and how they behave. So, you know, calcium oxalate is an organic mineral, calcium carbonate's inorganic, but there's a lot of similarities, and you'll see those as we go. And then in this Cook Medical from this year, look at the distribution. It's not just globally homogeneous, right? It's really the epicenter is here in Asia. And another thing to say about kidney stones, they can kill you. So of the 12% who have kidney stones, there's a mortality, actual death rate that's down around 2%, and it's growing as we go. So it's one of those things where people talk about kidney stones, hopefully you've never had them, and they are deadly serious ailment that needs to be dealt with, and right now we don't have a whole lot of good solutions. When I started the work, I was told a few things. Bruce, it's very nice that, you know, I've never met a geologist before, and it's very nice we've met you. But you know, there's not much to do on kidney stones. First of all, they never dissolve in the human body. Just remember, they never, when I hear those things, those are the things I want to study, because how the heck do you know they don't dissolve, right? They don't know. So they never dissolve in the human body, right? Another thing is they're homogeneous lumps that don't tell you anything, right? No. And there's many other aspects of them that really don't tell you anything. So really what everyone's been on is a search and destroy of this crystalline aggregate that doesn't tell you anything. And what they've done is elegant biology work, histology sections, TEM, a tiny bit of bright field transmitted light work, but they've done these incredible factor analyses, they call them, where you have different pathways of genes and proteins and cell wall debris, and you have inflammation. And the biology is worked out beautifully, right? But the rock record is the endpoint in which everything's recorded and encapsulated. And guess what? They never talked to anybody about that. Now, to start this journey, there's a very important fellow, Nicholas Stino. And for those of us who are aficionados and disciples of the world of sedimentology and stratigraphy, Stino was a medical student in Copenhagen. And in 1669, and I'm not trying to make everyone feel bad who's trying to write a dissertation, but it's pretty impressive. This was it was the prologue to his PhD, MD dissertation at Copenhagen. In that not in the dissertation, in the intro to it, about 10 pages, he identified the first salivary ductland. He did the law of constancy of interfacial angles in crystals as they grow, long before Miller. He did the law of superposition where, among other things, there's other aspects of this, but in an undisturbed sequence of sedimentary deposits, rocks at the bottom are older than rocks at the top. And then he also did something that we take for granted now, but he confirmed the biological origin of fossils. If you can believe it, like shark teeth that were collected from the White Cliffs of Copenhagen, they were interpreted to be the result of pressure and temperature, and that biology had nothing to do with it. And so he did all this, and I just wanted to point out that as soon as he graduated, he left science and medicine. He moved to Germany. He left Lutheranism and became a Catholic bishop. And then they sent him to lead the counter revolution, where after the Protestant firestorm burned down all the churches of Northern Europe, then the Vatican sent in the Vatican's troops, and they burned down and killed a lot of the people who'd killed and burned down the Catholic churches, and they reinstated a lot of Catholicism within Northern Europe after the Protestant firestorm, and he was the mastermind of all that. And then I just want to point out one more ironic thing. It's beyond ironic, is that he died of kidney stones. And so this poor fellow at the age of 46, that's his kidney stone. And somehow, I don't know if this is a girlfriend, a liaison, some kind of relationship that we don't know too much about, got the kidney stone out of his body, cleaned it up, and wore it as a brooch. Whoa. So I'm calling this Back to the Future. Oh yeah, that's how it works, okay? Now, what I'd like to share with you is I've been working with the Mayo Clinic on this for 10 years. And the Mayo Clinic made an 18-minute documentary, and that's where all the science is put out. They made a 2-minute trailer where almost none of the science is, but it's only 2 minutes long. But it's just a good segue to show you this quickly, and hopefully this is going to work okay. Can you hear it?
We probably see a couple of patients a day at the emergency department with kidney stones. I have had several women tell me that this hurts worse than labor.
What we're trying to understand on many different fronts is why some people get kidney stones and why some people don't.
I've always, as a geologist, wondered, what's the geology of a kidney stone? The similarities between the human kidney and hot spring environments, such as those that occur in Yellowstone, are simply remarkable. Mayo is the kind of place where it's not a harebrained idea to have a geologist knock on the door.
Where nephrologists have worked most closely is understanding the physiology or how the stones develop.
I haven't ever met a rock that hasn't crystallized and dissolved and crystallized and dissolved and crystallized and dissolved. That kidney stone is a dynamic history of both crystal growth and crystal dissolving.
If we can figure out what's leading to the rock growing within us, we hopefully can interrupt that with either antibiotics or a drug that targets a specific protein that might be leading to the accumulation of rock.
I think it'll be a game changer really for kidney stones.
It's rare to get people like geologists and medical doctors to sit down and do something serious together in terms of some research. But when that happens, magic happens. All right, so if you want the science download, you can get for free onto the Mayo website, and it's an 18-minute video. But I'm very appreciative that Mayo has gotten behind the work and we're now developing new therapies around it. So now what I want to do for the time left is just show you what I mean. How are the geosciences making such a big impact in the world of, in this case, kidney medicine? And I'm going to start off with, again, the experts in UTCT are sitting here in the front row. But on our instrument, these are 3-micron resolution MicroCT analyses of calcium oxalate kidney stones. The left two are what we call COD, calcium oxalate dihydrate, and then the right one, calcium oxalate monohydrate. And if you ever had any questions about why on earth are kidney stones so painful, I think all you have to do is look at these calcium oxalate dihydrate crystal terminations, and then think about that lacerating, slowly, cutting in 1,000 places your urethra. Oh, sorry. I think that we have a way to understand why this is so painful and why it can be so deadly. So we're having discussions today. I start everything I do now with MicroCT, the 3-dimensional contextualization and the ability to work through from that. Now, there are these statements, they're kind of that feel good around a campfire statement. Oh, Bruce, it's nice you're a geologist, but kidney stones never dissolve. So we went in and quantified the porosity that we found in 300 kidney stones. And on average, here's the MicroCT of an average kidney stone. The green and the red are the average porosities that we found. And on average, by volume, in vivo, in the human body, with nothing else being done, 50% of the stone is dissolving, then growing, and dissolving, and then growing. So it's just the opposite. And that history, the paragenetic sequence, gives us a frame of reference with which to develop brand new therapies that haven't even been talked about. But another thing that I just wanted to point out is that what's been remarkable is, I've been so thankful to have reporters and really engaged reviewers for our work, but the discussions with them have also really changed how we do our work because of their insights and the kind of questions they were asking. And one of them was that another nice story I was told that kidney stones never merge together to make a bigger kidney stone. And then, oh, wait, did you ever look at the stone? So this is unbelievable. This is just one of 300 stones. Almost every stone, what happens is these are individual stones that came together, dissolved, merged, and sutured, and became a larger kidney stone. That's a huge, that's an important segue, again, as a target for where we should do therapeutics. Another one is the amazing sedimentology and stratigraphy, right? I mean, I still get goosebumps when I see this, right? It's just, I love doing, everything from sedstrat geochemistry to structural geology, and, I can't tell you how critically important field observations have been for me over the years to be able to deliver this into the operating room. I mean, the field component is astronomically important. And so you can kind of get an idea here. Look at those cross-cutting relationships. This is calcium oxalate monohydrate. Those are 140 nanometer resolution layers. Those layers are made of proteins, mucus, cell wall debris, extracellular polymeric substances, all kinds of organic material that's being formed. Actually, I was told when we started that the kidney's sterile. And so we went in, took samples, and we had the first paper to sequence out and identify. We found 30 different types of microbes living in the average kidney, right? So all these kind of things, people used to say that Yellowstone hot springs are sterile, and it's the birthplace of life, right? It's the mecca of life. So again, to the students and postdocs and things, when you hear dogma told to you, that's when you question, you go right to the heart of the matter. Someone has just defined for you a really valid way to do research. Another thing I want to point out to you, there's people in here who know how to look at faults and things much better than I do, but look at the faulting. So this is faulting inside the kidney on the kidney stone of a patient in Mayo Clinic. And so we think that this is being driven by fluctuations, wild fluctuations in the salinity of the urine. And, they have found that gypsum crystals and things actually can be driven by salinity change faulting. So anyway, I just want to give you a feel for how remarkable and this is using everything we do in the geosciences to interpret this. Now let me just walk you through this. Here's the stone that came out of this patient at Mayo Clinic. We did the MicroCT, and we spent hours and hours looking at virtual cross sections, and we found just the right orientation, just the right cross section, because we wanted to find a cross section that gave us the entire run of the history of crystallization and dissolution and other paragenesis. But you have to have the right orientation cut to actually capture that. Here's a bright field transmitted light image of that. Look at that, right? Look at the stratigraphy, molds of dissolution. You go through different genesis of, this is not a homogeneous lump of rock that tells you nothing, right? It's just incredible. Then you hit this with 3 wavelengths of light, of ultraviolet light, to generate autofluorescence. This is the natural fluorescence of the cell debris, the molecules, other things that are trapped in those layers. And this is the same place, and so all the colors represent fundamental changes in the chemistry and the physiology of the kidney at the time they were laid down. So it's incredible how much information is here. So we took it and put it all together. Here's the autofluorescence image, and we made a graphic novel, out of the paragenetic sequence. And so this, plus analyses of the isotopes and the chemistries and the molecules and everything else we found here, allowed us to reconstruct this. The first 4 top ones, the pink is the renal tissue, the kidney tissue, and all these little flecks and things are crystals that are growing in the urine. This is a cross-section of a tubule, and then the crystals start growing into the flesh, and then some of the crystals are transported, and then we've color-coded them according to the crystal colors in the autofluorescence, and then you grow them, and this makes a plug, so they actually call this Randall's plaque. And then when it grows along a duct of the Bellini, the duct of Bellini and the loop of Henley are these morphological, physiological parts of the kidney. And then we were able to reconstruct that history. So what this is a roadmap for the future of therapeutic development. Because now, these are all things we can intervene with medicine, antibiotics, chemistries, dietary changes, hydration, genetic propensity to grow stones. This is what was missing. And this is geoscience, squarely in the heart of medicine. And another thing we see over and over again is, again, well established within the geosciences, the idea that microspherules, small spherules, actually coalesce together to make planar, beautiful euhedral crystals. And Oswald ripening is a part of that. And we see that here. Here's the calcium oxalate dihydrate being synthesized from spherules composed of amorphous calcium phosphate. And then those spherules that are here are coming together. So, in the past when I'd see that, I'd say, poor crystal, it's dissolving, it's going through diagenesis. Not at all. It's actually that crystal is growing from the process of the coalescence of the spherules. And then this is an agate that we collected from Kazakhstan, from a volcanic field. And look at in the agate how beautifully the spherules are shown to line up, and then this was kind of frozen in the process of spherules becoming planar features, and this is well documented in the literature. Another thing is I want to really emphasize is, and we're now seeing this in the kidney stones, we're seeing the corals, and we're seeing it especially in the hot springs. Kind of the world of modern and ancient biomarkers are going to be, at least for what we want to pursue, are the crystalline architecture, the sed-strat composition of the deposits, the crystal growth rates. You saw that amazing time lapse, and you understand what time has to mean for the kidney stones. A lot of the stones are one year to 2 years in duration. And then, but especially important, the nanolayering. So as an example, this is a a dolomite from the island of Curacao. It's in the Cerro Formation from my PhD work. And we're seeing exactly these three things, crystalline architecture, growth rate, and the nanolayering being played out even in a Pliocene age dolomite, right? So everything's there, including sector zones. We see, look at the sector zones in this thing. It's just beautiful, right? Then another thing is, when I started, there were only two worlds to form stones. One of them was the loop of Henley. So here's the kidney, and this is the renal calyx. It's the kind of the cavity, and then you have a bunch of these triangles. They're called pyramids, and each pyramid has 1 million of these tubules. So each renal pyramid has 1 million of these tubules, and the artery comes here. It feeds the blood through the glomerulus, and it goes through a long loop they call the loop of Henley, and the urine comes back, and it goes down through a collecting duct, and it goes out into the renal pelvis and goes down through the ureter, okay? And so this is the world I started with, and they said there's only two places you can grow stones, down here and down here, in the duct of Bellini and the loop of Henley. And as a geologist, I said, well, how about all the rest of this outcrop distribution, right? They never looked, right? So it turns out that we've been able to show that kidney stones grow every place in this system. And I'll just show you quickly, we just had a paper come out a few weeks ago, and we call it renal macrophages and multinucleated giant cells. I'll tell you what those are. But they operate like the ferrymen of the river Styx. So in Greek mythology, if you die, you have to be buried with a coin in your mouth, so that when your poor departed soul comes down these tortuous pathways along these canyons that lead to Hades, you have to board a ferry that is modulated in terms of the rate of loading of the ferry by Charon, C-H-A-R-O-N. And everything depended on Charon, and then this is the river Styx, and your soul has to get on that boat. Otherwise, you go into purgatory. But you're trying to get to either heaven or hell on the other side of the river Styx. And so everything depends on that, and the loading of the boat, the rate at which the souls come, that's all Charon's deal. So what we did was we started finding that there were these incredibly important cells in our body. When we get inflammation, those cells go to the inflammation, whether it's in your heart, your elbow, wherever the problem is. And part of them are called macrophages. They're large cells or giant cells. They're great big cells. And I just had a question. Can you ever have a cell grab crystals, incorporate the crystal, and then transport it someplace else and dump it out? And the answer was, we don't know. So we started using our microscopes looking at this. The left-hand side, I know there's a lot of detail here, but here's urine, here's mucus, here's the membrane around our tubules, and then here are macrophages and giant cells. And these crystals are calcium oxalate dihydrate and calcium oxalate monohydrate. The bottom line is, when our system's working, like hopefully everyone in the room's in this boat right now, these cells capture crystals and they transport them down, and then they slowly release them as crystals into the middle of the kidney, and everything's good. The right-hand side, all hell literally has broken loose, right? The entire transport mechanism breaks down. Nothing's moving through here. Charon takes a vacation. The poor souls are cramming onto the ferry across the river Styx, and it sinks, and all kinds of calamities, and bedlam is going on. And what happens is all these cells that are there to help, it's like trying to save someone on scuba, right? You never go up to a struggling person, because that person can struggle to try to grab you and pull you under, right? So all these crystals are trying to save themselves on scuba, and they're creating catastrophe. And then the giant crystals, I don't know if you've ever seen it, but people actually have a big lump on their back if the kidney stones get too large, and some of the kidney stones have been this big, and they're called staghorns, and they fill the entire kidney. So this is serious business, and this gives us then an ability to look forward. So here's our eternal triangle. Gives free energy, all the diagenesis and paragenesis I told you about, and the stair steps are solid phase transformations. The dotted lines are going from solution, in this case urine, into the deposition of the crystals, and then we have now for the first time petrography, chemistry, and organic chemistry to go with it. So the point is that this is what we need to build now for all the different cases of biomineralization in the human body. And now what we're doing is going into both the solid phase transformations and the direct crystal precipitation from solution, and deciphering targets for therapeutic development to treat kidney stones. And then I'm almost done. I'm just going to show you one last thing there's another big problem. It's called cardiovascular calcification. So I don't know if you notice it or not, but every year 18 million people die on planet Earth from cardiovascular disease. 18 million. And every year within the US, one person dies every 34 seconds from cardiovascular disease. $229 billion are being spent on this. Globally, it's hard to even fathom the impact. More people die from cardiovascular disease than war, famine, disease, everything else put together. And so you can see the distributions. Again, Russia and Asia are real hotspots, Central South Africa, and then the US is kind of running from the south then upwards through this corridor. So, but the problem is, people have known that there's another calamity happening, and that is calcification. So this is a human heart from 2011, cardiovascular pathology. That's the cadaver heart. This is a sliced heart, and the colors you see there, that's calcium-rich minerals. They've never analyzed the mineralogy, never done the chemistry, never looked at the stratigraphy, never made a paragenetic sequence, have no idea of thermodynamic recrystallization. This is killing people, and we think that maybe every year 2 to 3 million people are dying on planet Earth from cardiovascular calcification. And we have no idea what's going on. The doctors have done the same thing, some beautiful work. This is a reconstruction of a MicroCT, but most of the work's been done on tissues, a little bit of reconstruction, but mostly it's been in the biology, right? They have all these metabolic pathways, genes and proteins and enzymes, but they have nowhere to go with it, right? Because they've never looked at the end product of the calcification. So I've been working with the Cardiac Arrhythmia Center out at UCLA. UCLA Health, the Ronald Reagan Medical Center, is the #1 heart disease hospital in the world. So that's why I decided to work with them, because they have such a high throughput of patients. And so this is a cadaver heart, someone who was 52 and died from cardiovascular calcification. This was sent to us at Illinois. We put it into holding chambers, and then we put it into our MicroCT. And then here's what the analysis looked like. Here's a low-resolution scan where we can still see tissue, and you can see the hot spots. That's calcium-rich calcification. That's all we know. I mean, we can make hypotheses, right? Hydroxyapatite, calcium apatite,